What I want to do today is to talk a little bit about Shannon Point and some of the kinds of things that go on there, and tell you about how you all can get involved in either taking classes there or some of the activities if you're interested. And then I'm going to spend a little bit of time talking about the kinds of research that I've been doing at Shannon Point and how Western students have been involved in some of that research. So for those of you who do not know where Shannon Point is, we're up here at Western in Bellingham. Shannon Point is located down here in Anacortes, right next to the ferry terminal. This is an overhead view, so here's where you pick up the ferry to go out to the San Juan Islands. Shannon Point is located on this piece of property right here. It's about 78 acres of a wooded biological preserve, and then we have about 1,000 feet of shoreline that's accessible from the marine station here. This building here is a residence where students live, and I'll show you a few more pictures of what things look like down here. This is the main Shannon Point building with offices, laboratories, teaching laboratories where classes take place. And then this is our lecture hall, so if you're taking a class down at Shannon Point, you'd be spending some time in there. And these are the residences. We have 12 rooms at the undergraduate residence up in the woods, and so we can house about 24 people. That means if you're taking a lot of classes at Shannon Point and you don't want to commute back and forth all the time, you could actually live here. And we have a lot of summer programs where people are staying at the lab for the full time. So the undergraduates in those programs are staying in these residences. And the residences include some cooking facilities and a commons room for meetings. We also have this house where graduate students often live, or where we house visiting scientists who are coming to the lab. What takes place at Shannon Point are really three different things: research, education, and outreach. And I really shouldn't say that they're different, because at Shannon Point, all of these things are very integrated with one another. So we do a lot of research there, but all that research involves undergraduates and graduate students. And even our outreach programs are built on the research and education that goes on there. And so just to describe a little bit of the kinds of research that we do at Shannon Point, there are 5 of us that have sort of our home base at Shannon Point. 3 are faculty members at Western, 2 of us are senior research staff. And we do a lot of studies on things like organismal biology and ecology, on plankton, on seaweeds, seagrasses, and marine invertebrates. We also study community and ecosystem ecology and ocean and organismal chemistry. And in addition to the 5 people that are home based at Shannon Point, there are also about 15 affiliated faculty members, people who have their faculty appointments up here in Bellingham, but who either spend time teaching or conducting research down in Anacortes. And that allows us to expand some of the areas of expertise that we cover to include things like marine mammals and birds and fish and sediment biogeochemistry and physical oceanography. So we have a pretty good range of topics that we work on, everything from biology to chemistry to physical oceanography. And hopefully by next year we're going to add some, at least one geologist to that list. The kinds of programs that take place at Shannon Point involve Western students from before they even enroll in their freshman courses all the way to graduate school. So kind of taking those in order, one of the programs we have here that we offer is called Marine Science Scholars. How many in here were Marine Science Scholars? Do we have any? Oh, we've got a few. This is sort of like marine biology boot camp. You show up at Shannon Point 2 weeks before you're going to start your freshman courses at Western. You take classes and you go out in the field, and you even get to find out about careers in marine biology. This is a field trip to Taylor Shellfish, and this involves about 20 students. And these students go on to take classes together for the next couple of years as freshmen and sophomores at Western. We also offer a lot of Western courses at Shannon Point, particularly during spring quarter, but we're now having more in fall and winter quarter. This is a list that I put together of ones that I knew about. Like all departments at Western, courses kind of come and go. They're not all held every year. But typically, we do have some that seem to be offered every year, including marine algae, marine invertebrates, marine ecology. I think oceanography is also there every year. And the courses at Shannon Point are a little bit different than the ones up here in Bellingham. Because the way that they're organized is they typically run all day for 1 day a week instead of being, Monday, Wednesday, Friday. Marine algae is all day Tuesday. So you start around 8 in the morning, you go to 5 in the afternoon, but you only have to drive back and forth on Tuesdays. And so that makes scheduling a little bit easier for folks and also reduces the commuting time. And lots of people carpool back and forth too, that kind of helps out. So if you're interested in taking courses down at the Marine Lab, check out ClassFinder and see what's being offered. We also offer all sorts of internship programs, usually in coordination with departments on campus, particularly environmental sciences and biology. And if this is something that interests you, you pretty much have to contact a faculty member or one of the researchers at Shannon Point and work out things individually. Typically, I have 1 or 2 interns from environmental sciences working with me every year, and then they use that experience to take the 498 internship credits that they need to graduate. So there are opportunities for Western students to do marine research. And then coming soon, I suspect most of you have heard that Western's soon going to be offering a marine science degree. This was just approved last year. The money's just starting to come through, and there are now decisions being made about what this degree is going to look like. It'll probably be starting in 2019, so if it's something that interests you, keep an eye out for that. We're expecting a lot more courses at Shannon Point and a lot more research opportunities as a result of this new degree. So those are sort of the opportunities for undergraduates. Shannon Point also is very much involved with Western's master's program. The MESP program, the Marine and Estuarine Science program that's run through biology and Huxley, their students can come down to Shannon Point and do their thesis research at the Marine Lab. And typically we have anywhere from, say, 3 to 15 students working on a whole variety of different topics. They might be doing lab work like you see here. They might be out in the field diving or on the boats. So it's a really great place to do your master's thesis. So those are the programs that we have that are specifically for Western students. We also are research experiences for undergraduate site, and this is a program that's funded by the National Science Foundation. And every summer, 8 students come from all over the country to Shannon Point. They get paired with a faculty member and they spend 9 weeks doing research and learning about what it's like to be a marine scientist. And typically every year 1 or 2 of these students are from Western, so if this is something that you think might be really interesting, you want to see what it's like to do intensive research for a summer, you can apply to this. Unfortunately, we're not running it next summer. But we are not the only REU site in the country. The National Science Foundation funds a whole lot of these REU sites, and it's not just marine science, it's all sorts of different topics. There's a chemistry REU site here at Western. There's some in forest ecology, tropical ecology. If you're interested in this kind of thing, just Google National Science Foundation REU, and you can see what all the different sites are. And apply to those programs. And these are really great programs for getting some real hands-on experience and learning about what it's like to be a researcher. So those are the educational programs we offer at Shannon Point. We also do a lot of community outreach, and this is just a list of all the different programs, and you can see there's a long list. A lot of them are youth programs. They're targeted towards K-9 students, so we're involved with the local schools. And there are opportunities for you as Western students to become involved in this. If this is something that you think you might be interested in, you want to get some experience in, I shouldn't say all of these, but at least some of these programs are taught by Western undergraduates and graduate students. And we're in the process of hiring an outreach coordinator right now. So not a lot's going on fall quarter, but come winter quarter, I think we'll be looking for some folks to help out with these programs. And I think some of them are actually paid positions, so you can get a job doing this. So that's something else you can do down at Shannon Point to get some experience. So that's a little bit about what happens at Shannon Point. I was going to spend the rest of the time talking about my research program and what it is that I've been working on. And a lot of what I do involves undergraduates, mostly a lot of Western students, but also students who have gone through the research experiences for undergraduate program. And I thought I'd talk today about a research project that I've been working on for quite a while, partly because it got started by an undergraduate research senior thesis, and it's actually kind of taken off and gone in a lot of different directions. I'm a marine chemical ecologist, and what that means is I'm interested in marine organisms and these weird chemicals that they produce that affect their interactions with other organisms. So if you can imagine a plant, like a seaweed growing on the shore, and along comes something that wants it to eat it, it can't run away and it can't fight back because, you know, it's a seaweed. And so what a lot of these organisms do is they produce these weird chemicals that taste bad. They might prevent pathogen growth. And so this is the kind of thing that I'm interested in. Sometimes these are called molecules of keystone significance because they're produced in fairly low amounts by just a few organisms, but they can have some pretty profound impacts on marine ecosystems. And so I'm interested in how these regulate interactions both intraspecifically and interspecifically, you know, within and among species, how this translates into changing the structure of marine communities, what happens if these things get released into the air or the seawater, and then how do organisms decide how much of these things to make? Because these things take energy to make, they take resources, and so basically the idea is that there's a trade-off between making, say, defensive chemicals and making babies. And if you want to pass your genes on to the next generation, what's the best strategy? Making lots of babies or making sure you don't get eaten first before you make the babies? And how do you regulate this? And how does the environment affect this? How does climate change affect this? How does just natural environmental variation affect how much of these get made? And how do things like nutrient pollution affect these? So the system that I want to tell you about involves green tides, and for those of you who haven't heard of green tides before, these are harmful seaweed blooms. The seaweeds that I'm working on grow naturally in the environment. They make good food and habitat for lots of plants and animals, but sometimes they get out of control, and that's when they become harmful. We like them in moderation, but when they go crazy, that's a problem. And these harmful seaweed blooms are occurring all over the United States. So the algae that I work on, the seaweeds, are green seaweeds. You can see we have green seaweed blooms all along the West Coast. They happen in the Northeast. They happen down in the Gulf of Mexico and on the Florida coast. So they're all over the place. They're also happening all over the world. And this is sort of our poster child for a green tide. This is the city you're looking at here is Qingdao in China, and you can see this whole embayment just filled up with green seaweeds. There's a story behind this. This picture was taken in 2008, and if you think back to 2008 and what was going on in China that summer, that was when the Chinese were hosting the Summer Olympics. If you remember, they were trying to make a fantastic show of it, have everything go really perfectly, and show what a good job they could do with the Olympics. And this whole batch of seaweed just washes up into this embayment about 2 to 3 weeks before the sailboat races were supposed to take place right around here. And of course, this was kind of embarrassing for the Chinese government because they were trying to make everything go so smoothly. So they mobilized the military. They got lots of volunteers from the local city. Everyone went out there with big burlap bags, bagged up all this seaweed, hauled it off to a local farm, and the sailboat races took place as scheduled, and they apparently went smoothly. But this big green tide bloom has been showing up every year since 2008. 2008 was the first occurrence, and it was estimated that this bloom was about 100 million tons of seaweed. So this is a lot of seaweed. This is the biggest green tide bloom that I know of in the world. We also have green tides here in the Salish Sea. They're fortunately not as big as the one in China, but here are a couple places near Seattle where we've got green tides going on. This is Ship Harbor, located on the other side of the Anacortes ferry terminal from Shannon Point. You see a lot of green seaweeds showing up there. This is another view of that Ship Harbor bloom, and you can see all this green in here. There's some eelgrasses kind of mixed in among them. These are all the people waiting for their ferry. And you can just see that the green algae are just overgrowing all these eelgrasses. And eelgrasses are really important habitats for a lot of organisms, so we don't want these big green tides overgrowing them all. The blooms that I study, the ones close to Shannon Point, one called Ulvalactuca and one called Alveria obscura. And if you go out and look at these in the field, they kind of look like, almost like green pieces of paper. They're about that size. And they look very similar to one another. Actually, they all kind of look like this. This is a real pretty one that somebody's pressed. But if you take them into the lab and section them and just slice them really finely, ulva has 2 cells, so you can tell it apart from Alveria that has these long rectangular cells. And so for every piece of seaweed that we've worked with, and we've probably worked with tens of thousands of them, we have to slice them open and check and make sure they are what we think they are. And this whole research project that I'm going to tell you about got started because of an undergraduate project at Seattle Pacific University. There was a student there named Amara Nelson who was working with Dr. Tim Nelson, not related. And what Amara was interested in is how come there are all these seaweeds out there and nobody's eating them? You know, these are called sea lettuce. You know, they sound like they ought to be nice and tasty. And she was also interested in whether or not they preferred ulva over Alveria, if the herbivores could tell these two apart. And so she did a lot of feeding experiments giving all these different grazers choices of these two seaweeds. And what she found is that everybody, given a choice, would rather eat ulva than Alveria. Some of them really don't like to eat ulva, but if they only have these two, they're going to pick ulva. And so Amara did this as her senior thesis project. She finished up at Seattle Pacific University and decided she wanted to go on for a master's degree. So she came knocking on my door and said, you know, I know you work on this kind of stuff. I know what I want to do for my master's thesis. I want to find out why nobody likes Alveria. We kind of know there's a chemical in there somewhere that things don't like the taste of it, and I want to figure out what that is. And I said, oh, okay, it sounds like a really interesting project. I know how to do this, but this isn't easy to do. This is like looking for a needle in a haystack, because these seaweeds are made up of thousands of different chemicals. We can extract them all out, but how do you know which one of those thousands of chemicals is the one that tastes bad to these herbivores? And to do this, we used a process called a bioassay-guided fractionation. And so imagine that this bar here represents all these thousands of different chemicals, and each different color is a different chemical. You take all these chemicals out of the seaweeds, and you're trying to figure out which one is the one. You know, like the analogy of looking for a needle in a haystack really kind of applies well here. Well, if you think about this, if you're looking for a needle in a haystack, wouldn't it be easier to find that needle if you could split that haystack in half and have two smaller haystacks and say, the needle's in this one, not this one, so we can ignore all this stuff and we can figure out, which of these two smaller haystacks it's in. And that's essentially the idea behind a bioassay-guided fractionation. Whoops. We get all these chemicals out of the Alveria, we split it into two groups using a separatory funnel. This group tends to have the more nonpolar chemicals, this one has the more polar ones. The ones in the middle kind of split in between these two, and then we use a bioassay to say, okay, which one has the chemical? And the bioassay that we used involved feeding sea urchins little agar cookies that we made. So here are our agar cookies. For those of you who aren't familiar with agar, it's kind of like a really stiff jello. It's the same stuff you grow bacteria on in your microbiology class. We grind up kelp and freeze dry it, and we put it into the cookies because urchins like kelp. And then when we're making up the cookies, we can put these mixes of chemicals in them. So we can say, put all the chemicals in the treated one and leave them out of the control one. Let the sea urchins feed for a little while, measure how much they eat, and then we can just subtract the amount they eat of the ones without the chemicals, or the ones with the chemicals from the ones without the chemicals. Now let's imagine that that you put chemicals in there, but they aren't the bad tasting ones, and the urchins don't care about them. If you take the amount of the controls and you subtract from that the amount they eat of the treated ones, what do you think the number's going to be? Anybody? If they don't care, if they like the two of them equally, it's going to be 0, right? You put chemicals in here that don't taste bad or they don't care about them, they're going to eat about the same amount of the two agar cookies. What if you put the bad tasting chemicals in the treated ones? What's this going to be? Is it going to be a positive number or a negative number? Okay, so the bad tasting ones are in here. So how much are they going to eat of this? A lot or a little? A little. How much are they going to eat of the control? A lot. So you got a big number for C, a little number for T. Okay, right? If the bad tasting chemicals are in the treated one, this should be a large positive number. If there's something that actually makes the cookies taste even better, then this is going to be a negative number. It'll go the other direction. And so this is what Amara did in her thesis, is she went and she took all these chemicals out of Alvaria, she split them into 2 groups and ran 2 feeding assays, one with the polar ones and one with the non-polar ones. So this is essentially C minus T right here. And what we're looking for are large positive numbers. And we run some statistical tests on these and we find out that it's the polar ones that they don't like to eat. So then what we do is we go and we subdivide this even farther, okay? So we know these non-polar ones really don't matter, even though they weren't as tasty, quite as tasty as the controls, but they really dislike these polar ones. So the next step is to take just the polar ones, because we're going to ignore those, and we're going to subdivide these even farther into 11 different groups. And we do this using column chromatography, chemistry technique, and then Amara went and did 11 more bioassays. And this is what she found out. So these are the next 11 bioassays, putting each of these into the treated cookies and comparing them to controls. And you can see that they don't like the ones down here, the really polar ones. So what we do at this point is we take all of these, we combine them, oops, and somehow here we go. And then we used a technique called high performance liquid chromatography. And so we took those and we divided them into 20 pieces, okay? So we've kind of tossed out all these because we're not interested in them. We're only interested in this batch, and we take this batch and divide it up into 20. And then Amara goes and does another 20 bioassays. These things are not easy, by the way. She did a lot of work. And what she found out is that only 2 of these fractions from the HPLC tasted bad to the urchins. The rest are kind of up and down, but they weren't significantly different from 0. So we know that the chemical that the urchins don't like are in these 2. So at this point, I call up Dr. Vivian over in the chemistry department. We call up the folks at SciTech. We all get together and say, okay, what's in these 2 right here? We've got it narrowed down to a really small group of chemicals. And it turns out that 2 things were in those. One was a chemical called dimethylsulfoniopropionyate, or DMSP. And I had worked with this chemical a lot, and I knew it wasn't something that tasted bad to the urchins. In fact, it tasted good to them. They actually liked the taste of it. And the other thing in there was dopamine. Now, I suspect most of you have heard of dopamine, right? Yeah, you all have dopamine in your brains. It's a neurotransmitter. You use dopamine as a hormone. All animals do. But it turns out Alvaria has tons and tons of dopamine in it. The concentrations in Alvaria are like 1 billion to 1 trillion times higher than the concentrations in your brain. And these things are loaded with dopamine. But even at that point, we weren't absolutely certain that it was the dopamine that was making the algae taste bad. So what we did is we call up the chemical supply company, say, hey, I want a bottle of dopamine. They mail me one. We take some dopamine. We put dopamine in the cookies. We do another set of assays. And we do it with all of the herbivores that we used before. And nobody likes the taste of dopamine. So this is how you find out what chemical it is in something that has some effect. You can do the same thing if you're looking for a chemical in a sponge that causes cancer cells to stop growing. You do the same sort of process where you just start with everything you can get and narrow it down to figure out which are the ones that you're interested in. So this is a really nice method for doing this. So we find out that dopamine is in Alvaria, and it helps make the algae taste bad. Well, it turns out dopamine is also in a lot of other plants. In fact, lots of plants produce chemicals that are the same chemicals that you use as neurotransmitters. This book just came out a few weeks ago. I was very pleased to see that they put one of our pictures of Alvaria on the cover of the book. Alvaria is the only seaweed we've found so far that produces dopamine, but lots of other plants do. Things that you eat, like avocados and potatoes and bananas, you're eating dopamine all the time. But the amounts are really, really small. The concentrations in these foods are about 1,000 to 10,000 times lower than what we find in Alvaria. So is this the only reason Alvaria is producing dopamine? It's to make itself taste bad. Well, it turns out maybe not. We've got some evidence, and this is a follow-up study done by another Western student who went out and just measured the concentrations of dopamine in Alvaria over time, and this was during one summer, and what we see is it goes up in the summer, it kind of peaks in mid-July and then comes back down. So when it's nice and hot and sunny, they're making lots of dopamine. Okay, what does that mean? We also, another student was involved with this. We did an experiment looking to see whether or not if you change the conditions of Alvaria in the lab, do they make more or less dopamine? So we thought that these plants making dopamine, which has nitrogen in it, if we gave them lots of nitrogen, they ought to make more dopamine. Turns out that's not the case. They grow really well, but they don't make more dopamine. But what does make the dopamine increase is if we give them lots of sunlight, or lots of light. So these dark green bars are ones that are shaded, and these light green bars had a lot of light, and the Alvaria starts making more dopamine. So why would it do this? Well, it turns out that there's a number of chemical reactions that dopamine undergoes if it's in solution. So here's our dopamine over here. If it goes into solution, it forms this dopamine quinone and releases these nasty reactive oxygen species, and then kind of cyclizes and forms this red-colored quinone. And eventually, what it does is it makes melanins. And melanins are found in your hair and your skin, unless you're albino. If you go out and spend some time in the sun and your skin gets darker because you get a tan, it's because the melanins in your skin have increased. So it looks like our seaweeds might actually be giving themselves a tan by making more dopamine in the hottest part of the summer. Another thing that kind of supports this is where the dopamine is in the cell. So these are our big, long Alvaria cells, the nice rectangular ones. One of my colleagues got some fluorescent anti-dopamine and stained these things, so the dopamine shows up as bright green, and you see it up here at the top of the cell and the bottom of the cell. They're out at the edges of the cell, which is where you'd want to put a sunscreen. You don't want it stuck in the middle where it's not going to do you any good. So that's what dopamine looks like inside of cells. It looks like it's acting as a sunscreen and a feeding deterrent. Well, dopamine doesn't always stay inside of cells, and that actually creates another set of problems and a lot of opportunities for undergraduate and graduate research. This was one of our study sites on Blakely Island, and you see all this Alvaria washed up on the beach. It's low tide, so the tide's gone out. It kind of gets stranded here, it dries out. Tide comes back in and starts rehydrating all this stuff, and it's the seaweeds are amazing. They can dry out and rehydrate. But one of the things one of my colleagues noticed is the water along the edge of the tide, as it's coming back in over those dried up Alvaria, starts turning red. And so I was out there one time and I collected some of this water and just filtered the sediments out. You can see this kind of reddish orange color that it turned. And if I go back to this slide here, where we see what happens to dopamine in water, see this red colored quinone? That's what we're seeing right here, is that the dopamine's coming out of the Alvaria and going into seawater. So this got us thinking, the quinones are pretty nasty chemicals. They kill things. Dopamine quinone is known to be cytotoxic. It's known to be genotoxic. It can cause cancers. And then these reactive oxygen species are also very nasty. You've always been told to eat things with lots of antioxidants. You know why you're doing that? it's to get rid of these reactive oxygen species in your body. So those are nasty. They mutate DNA, they damage proteins, they damage the cell membranes. So we've got these seaweeds releasing lots of dopamine into the water. There's lots of dopamine, quinone, lots of reactive oxygen species. This doesn't sound like a good thing, right? So what some of my, first of all, actually I'm just going to bounce ahead here. What some of my students have done is to expose a lot of organisms in the water to dopamine to see, does it really hurt them or not? And so we've taken other seaweeds and taken eggs and sperm from them and let the eggs get fertilized to form zygotes and expose them to dopamine, and we find out that that kills the zygotes. We've taken Ulva lactuca, the other species of green algae in the blooms, and grown it with dopamine, and it turns out it slows down the growth rate. We've taken crab larvae, these little zoeal stages of some of our local crabs, and exposed them to dopamine, and they start dying. Interestingly, if the crabs are a little older, the dopamine has no effect on them. So this looks like it's not a good thing for the organisms living in these waters. One of the other projects that one of our graduate students did is that she was wondering what happens if you're a larva and you're not in the dopamine for a long time, but you just get exposed to it for a little while. Say you kind of swim through a patch of it for 15 minutes or an hour. It turns out that that's not enough to kill the larva, but what it does is it affects its development. So what she did was expose these sand dollar larvae to fairly low concentrations of dopamine relative to what we're seeing the algae release for short periods of time, and instead of just seeing if they're dead or not, in fact they all lived, she started measuring characteristics. So this is a gastrula stage, one of the early stages of the larva, and she measured the archenteron, that's this really primitive gut here, and when it's exposed to dopamine it gets shorter. So it's affecting the development somehow. She took these later stages, these plutei larvae, and exposed them to dopamine, and their legs got shorter. And this might not sound like a big deal, but for these larvae, how much food they can take in is dependent on how long their legs are. So if they're making shorter legs, that means they're not feeding as well. And if they're not feeding as well, they may not be surviving as well. So this dopamine that's coming out of these seaweeds may be affecting a lot of other organisms. And so that's some of the research that we've been looking at here. So if we go back to my original slide, you can see here in this system we have a lot of the components I talked about earlier. We have these marine organisms making dopamine, a molecule of keystone significance. The growth of the seaweed is affected by nutrients, but the dopamine concentration and the growth are both affected by light levels, and the amount of sunlight is expected to change based on what we think the results of climate change in the area will be. We're expecting sunnier skies in the summer when these algae are growing, so we might expect to see more dopamine in them. We see that the dopamine is being released into the seawater and changing the seawater chemistry through the formation of reactive oxygen species and dopamine quinone. The dopamine is affecting interactions with a whole variety of organisms. It's preventing things from eating the algae, which might be contributing to forming these big green tides. And it's also, the release of dopamine is also affecting organisms that live in the area, and we don't know what the long-term effects of that would be. And then finally, this is a whole nother story and a whole nother seminar, but these plants are also producing a lot of small sulfur compounds that get released into the atmosphere. And there's some suggestion that maybe some of these sulfur compounds from these very large blooms might be affecting cloud formation, at least in very local situations. So these are some of the kinds of things that are happening with these seaweeds. And it gives you a little flavor of the research that I've been doing out at Shannon Point. And I'm going to finish up by thanking all the folks that worked on these projects. I had a lot of collaborators from different institutions, a lot of undergraduates who helped out with this, a lot of graduate students, and of course all this costs money, so I have to thank the National Science Foundation for funding it, and NOAA as well. And I thank you for your attention.